Hi, again. I know this is kind of awkward that I'm leaning in, but I still figured that you should actually see me at some time when I'm talking about these things. This is going to be an overview of the citric acid cycle. If you ended up taking a class in biochemistry or something else like that, this is going to be way more in depth than, or way less in depth than you would have actually had with that. I just want to hit a couple key points in this. But some of you probably haven't seen this in like multiple years. So pay attention because there are going to be things that we're going to revisit when we end up talking about some nutrition type things. But remember, when we're talking about this whole thing, we have this equation at the top, or as I like to call it, the equation for life, or cellular respiration. We have C6H12O6 plus 6O2, so this is going to be aerobic respiration. So aerobic, meaning oxygen, and this is glucose, or pretty much any type of sugar that we're going to have here. And there's different ways that you can make things into glucose so that you can shove it into wherever it needs to be. And then throughout a whole entire process that we'll talk about down in this part, we're going to get 6CO2, 6H2O, and a whole crap ton of ATP. Now, we do need a little bit of ATP to kickstart this whole entire thing off, and we'll talk about that in a bit. But in general, you get somewhere or another around 32-ish ATP after everything's all said and done. One other thing to remember throughout this process is that with the use of carbonic anhydrase, we're going to be able to, or that is the enzyme that is going to catalyze CO2 plus H2O into H2CO3, or carbonic acid, which will dissociate into an H plus and an HCO3 minus. That's a 6. That is not an H, even though it looked like an H. Plus 6 HCO3 minus. So that is going to be our carbonic acid, a bicarbonate ion, and then our hydrogens that are alongside of it. Shove a sodium in there and you have baking soda or sodium bicarbonate. And now you know that. So, but this whole entire thing, so this is the reason, this part of this is how CO2 travels in blood, and why your body is going to look at the amount of acid in your blood to determine how fast you have to breathe. We'll talk more about that later. But when we're talking about this whole process and how to actually get ATP, so the cellular respiration aspect of things, or how we end up getting actual oxygen out of things, then here's my pug. That's Olive. She isn't supposed to be down here, but she came down anyway. Now, you see her? That's it. Sorry about that brief interlude, but as we're getting back into that, this whole thing, when we're talking about cellular respiration and how to get ATP, or energy, out of glucose, really why we end up eating glucose or eating things in general, we are going to talk about this whole cycle, which is going to be broken down into a couple of different parts. Specifically, we're going to start out with the glycolysis step. And glycolysis itself, and I need to find a color that I can actually use that should be okay. So this is glycolysis itself. Is just splitting up of glucose. Glyco, meaning glucose, lysis, meaning split. So lysis as in breaking. So when we're talking about actual glycolysis, really that's just this first bit, going into 2-pyruvate. So taking a glucose, which is a 6-carbon sugar, and making it into pyruvate, which is a 3-carbon sugar. So 6-carbon sugar into 3-carbon sugar. And really what this is actually going to do is we take this glucose, we shift it into a fructose, add a couple of phosphates here and there, bing, bang, boom, split it into 2, and we end up getting our 2-pyruvate. That takes 2 ATP to actually do that whole process, but then with that, those 2 pyruvates, we add in a CoA and end up getting a couple ATP out and some NADH. NADH is going to be this whole thing that will act as a hydrogen carrier for a variety of other different things. So if you look at this, we have our 2 ATP in, 4 ATP out. So this glycolysis step itself is going to net 2 ATP. And if we end up actually bringing that forward a little bit more, we can use this pyruvate and we can go through some sort of lactic acid fermentation or ethanol-based fermentation, depending on, well, we can't because, you know, we're humans and we don't make booze, but yeast can. But either way, we'll end up getting a couple or 2 more ATP out of that whole entire thing. We can essentially take these pyruvates, shove them together with a couple of other different types of enzymes, and poof, you get pyruvate. Yeah, you add on you add on a CoA, and you end up getting 2 acetyl-CoA. And acetyl-CoA are essentially an acetic acid with this CoA structure attached to it. That also kicks off a CO2. Actually, it kicks off a couple CO2, and then you end up you also create 1 more NADH with that per each one. So I think you get 2 NADH because, you know, that would make a whole lot more sense. So we get 2 NADH, and we have 2 CO2 that are produced. Either way, this acetyl-CoA is actually going to be a 2-carbon structure. And that 2-carbon structure is going to feed into the citric acid cycle. The citric acid cycle is going to do a whole lot of random stuff to this acetyl group, and it's going to take that acetyl group and rip it off of different things, shove it onto other different types of things, do different types of things with it, take hydrogens out and shove them onto other things. So we're going to take a couple NAD and add some hydrogens onto it. We're going to take some FADHs and add some hydrogens onto it. We're going to make a GTP, which then can get converted into an ATP, because GTP actually is vaguely similar to ATP. It's just it uses guanine instead of adenosine. But essentially what we're going to do is we're going to get a whole bunch of hydrogens that we can bring to the electron transport chain. So this citric acid cycle is going to be the best thing that we can do. We're going to go through it twice. So we can say 2x, because we have 2 molecules of acetyl-CoA that we're going to feed into it. Okay, so that is also how we get this X2 over here, and then that is how we're going to feed into the electron transport chain. What the electron transport chain is actually going to be is in the different membranes within the mitochondria, the powerhouse of the cell. We're going to take these hydrogens and shove them across multiple times in multiple ways, so that we then can get hydrogens that will go back through, and that when it goes back through, it will feed into it'll create a hydrogen gradient, which will then feed into an ATP synthase. And the ATP synthase is this actual weird rotary machine that as it spins, it's going to provide energy. And it's going to spin because of the way that the hydrogen ions are actually going through it. Once again, if you really want to know more about that, take biochemistry. If you've taken biochemistry and I'm butchering anything that Dr. Terrell said, pay more attention to what she says than me. I'm talking about this more at a broader level, and this is my understanding of it, not necessarily the way that it actually is. I mean, it's a pretty close approximation, but still, one way or the other, this is how things go. So you shove a whole bunch of hydrogen ions out, you bring them back in through the ATP synthase, and then you end up getting this whole entire ATP synthesis type thing. And you can actually get this so that it pumps out somewhere or another between 26 and 30-ish ATP overall, so that once we get all said and done with it, we will create something along the lines of 30 to 36 ATP as net. And that is going to be a fair amount of ATP per glucose. Now, depending on what we actually feed in, sure, glucose makes sense, that's what it's built for, but there's also other different types of things that we can do to shove things into it. So if we have, say, glycerol, we can feed that in. If we have different types of amino acids or peptide chains, we can feed those in. So our fats, our proteins, We can feed in a variety of different types of things into this whole entire process to get ATP out, because that's what we care about. That is what I'm going to be talking about as far as this video goes. I hope you enjoyed it. I hope you enjoyed seeing Olive, my pug, and we'll see you later.